Most stem cell biologists, when the Harvard Stem Cell Institute was first started, were interested in using cells as therapeutics. But in the last 2 or 3 years, people's ideas about how to find therapeutics using stem cells has changed dramatically. And in particular, a lot of people are now focused in on using stem cells to better understand and model diseases, and then to use the stem cells to find conventional therapeutics, which is to say pills, small molecule kinds of drugs. The Therapeutic Screening Center was started in an attempt to help people do just that. So the center has a lot of collaborations with other members of the Harvard Stem Cell Institute, and in particular, a lot of our work revolves around doing very, very difficult stem cell-based assays focused on identifying specific kinds of therapeutics for certain kinds of diseases, particularly degenerative diseases or cancer, and also in deriving reagents that allow people to control stem cell properties. I came to Harvard actually having worked for about 10 years in a biotechnology company, and in the last few years of that company I had started working on orphan diseases. Orphan diseases are diseases that don't affect that many people, and hence there's not a big financial incentive for corporate entities to work in those areas. One of the diseases we work on is called spinal muscular atrophy, and it's the most frequent genetic killer of kids under the age of 2. And I felt it was very important for someone to work in this area. Because it's a motor neuron disease and motor neurons are very difficult to study, we were determined to try to study this disease using motor neurons derived from embryonic stem cells as an important component of our study systems. We carried out a screen that was designated to correct the problem with this disease, and the problem is that there's a protein called survival of motor neuron with slightly unknown function, but it is known to be necessary for motor neurons to survive, as the name suggests. The disease is caused when the level of this protein decreases too much. It needs to decrease to about 25% of normal, and if that happens, that actually leads to death within a year or two. So we were looking for small molecules, drug-like molecules, that could be used to treat kids that would cause an elevation of the level of this protein. We did a very sophisticated and difficult screen which involved growing cells in culture and imaging them using an automated high-throughput screening microscope, looking for any kind of molecule that would raise the level of this protein. We found about 150 molecules that raised the level of this protein, but we focused in on a small subset of the most drug-like molecules. And one of the ones that we just described in a paper that was published in Nature Chemical Biology acts as a kinase inhibitor, and the kinase inhibitor causes the protein to be stabilized and increase in amounts over time. And when that happens, when the protein increases, it does just what we were hoping for, which is to say it keeps motor neurons alive when they would otherwise die. So the next steps in this project are to actually start testing these compounds and the other ones that we identified in the screen in mouse models of spinal muscular atrophy, and then hopefully, ultimately, we would be very excited about trying to move some of these molecules into clinical development.